I would like to introduce you to three people, Nina, Robert, and Paul. They all went to the doctor with symptoms of depression and were prescribed the same antidepressant at the same dose. Nina takes the medication and she begins to feel better. Robert takes the same medication and nothing really changes for him. When Paul starts taking the medication, his condition gets worse. His heart races at night, he has headaches all the time. He gives it a few more weeks, but eventually stops taking the medication. Three people and very different outcomes. So here's the question that sits at the heart of my research. Why does the same medication behave so differently in different people? And what if those differences were not random at all? I work with the Estonian biobank, which holds genetic, health, and medical data from over 200,000 individuals. This represents about 20% of Estonia's adult population. In a recent study, we looked at 14,000 people who had taken antidepressants and asked, why do some of them experience side effects while others don't? We started with a gene called CYP2C19. This gene produces an enzyme that breaks down many commonly prescribed antidepressants, such as citalopram, sertraline, and amitriptyline. But not everyone's version of this gene works the same way. Some are what we call poor metabolizers. They cannot process the medication properly, so it builds up in the body. In our study, poor metabolizers had 50% higher odds of experiencing side effects than normal metabolizers. Others are ultra-rapid metabolizers. They break down the drug so quickly it barely has time to work. This means fewer side effects, but also less benefit. In the study, they had 17% lower odds of experiencing side effects. If we had sequenced Paul's genome, we would have known he was a poor metabolizer. He needed a much lower dose or a different medication to spare him from the side effects. Robert was the opposite. He needed a much higher dose or a different medication that would have given him the therapeutic benefit. Nina, she broke down the medication the way textbook says, and the standard dose was fine for her, and she got better. But these outcomes are not random. They are often a predictable consequence of how our body processes the medication, and information we could have had even before the treatment was prescribed. So we have two jobs. We first need to use the information we have, and we need to keep researching what we do not know. We know pharmacogenetics works, not just from this study, but from decades of research. However, in most countries, it's not being used. Even well-established scientific discoveries can take decades to reach the people they would benefit. In Estonia, we have created pharmacogenetic reports for 200,000 biobank participants. These reports can help the doctor with choosing the right dose and the medication for the patient. But now this needs to be integrated with the healthcare system so that doctors could actually use that information. And it needs to be extended for everyone, not just biobank participants. But there's a lot of things we do not know yet. There's a lot of what genes do that we do not know how it will affect the treatment. To keep learning, we need access to the data we already have, because currently a lot of it is locked away. And secondly, we need data that reflects the people who actually take these medications. Clinical trials remain our best test of whether a treatment works. And they're incredible, but they follow younger and healthier patients who have similar profiles and are only run for months rather than years. That's why we need real-world data where we can find out more about the people who actually take these medications for long periods of time. This data already exists. The harder question is, how can researchers access it? Health information is among the most sensitive information about us, and protecting it is non-negotiable. However, locking it away also has a cost. It's how variants get missed, how side effects go uncatalogued, and ultimately, patients keep being prescribed treatment that doesn't work for them. One example of data that stays locked away is doctor's notes. These can contain information about what doctors observe or what patients report to the doctor. And this can be details that patients themselves do not recognize or recall months later in a questionnaire. However, legal and regulatory barriers keep this information out of most research, even when privacy is protected. We were in a unique position to gain access to anonymized doctor's notes. Using natural language processing in a secure environment, we scanned nearly 100 million records to identify side effects. We found that 1 in 7 antidepressant side effect cases would have been missed if we only relied on doctors' questionnaires alone. And the true number is likely to be bigger because we only looked at a handful of most common drugs. This gave us a better picture of the range of side effects people have in the real world. And this was also the basis for our new finding. We looked at structural variants. So these are situations where part of the gene is missing. And we identified a case where the part is missing, meaning that the enzyme can't do its job properly and the medication builds up. We were the first to identify that carriers of this variant are at higher risk of antidepressant side effects due to significantly reduced enzyme activity. But what makes this interesting is actually that this variant is 10 times more common in Estonia than in other European subpopulations. This is why genetic tests matter from even small countries. Different people carry variants at very different rates. And when a population is missed, it means that variants like this one are excluded and missed. That's why we need genetic tests to include everyone in these tests to make sure that we can predict how the medication is processed properly. So this brings me to the main point. The goal is not medicine that works for the average patient. The goal is medicine that works for the patient receiving the treatment. So we have 2 jobs. We need to use the information we already know. The evidence is mounting. We now need experts and leaders to come together to bring pharmacogenetic testing out of the research lab into the doctor's office, so that the next poor does not have to stop treatment that was wrong for him from the start. And we need to keep researching what we don't know. There are variants with unknown effects because the data on treatment outcomes was locked away, or because these people carrying these variants were never in the study to begin with. This is also that more people could have a treatment experience like Nina's, because drugs work when it's the right match for the person.